guys, so here is one of several videos on solutions and diffusion. This is going to be the topic that you'll be seeing a lot of in the lab materials where another professor is going over the same sort of thing. I just wanted to give you a really basic introduction to it before you apply it in the lab situation. So what we're looking at here are solutions. And remember, this is anatomy and physiology for humans. So we're thinking in terms of what kind of solution we would have in our body. So we're always taking what we learn here and applying it to what's going on in the body. So we'll pull that together at the end here. But just to get a basic idea of what a solution is, it's just a mixture of things. Typically fluid, doesn't have to be fluid, but when we're thinking of in the body, we're thinking typically of fluid, like interstitial fluid, which is the fluid that is filling up all your tissues surrounding your cells, or blood, where you have plasma, which is the water part of blood, and there are things floating around in that. All of those things are solutions, and that's how they would be found in your body. Now, a solution consists of a solvent, and that's the thing that is dissolving the other things. You can think of it as kind of like the fluid part of it, whether it's a complete liquid or whether it's maybe a little bit more solid. Like, for example, bone, the interstitial fluid is quite solid, but the solvent is what has dissolved the other things. Now, what is dissolved within the solvent is called the solute. And I think the easiest way is to just look at this by example and just take salt water. So just a glass of salt water. It's sodium chloride, so NaCl, sodium and chlorine, plus water. That's a solution. And if you imagine the salt all evenly interspersed amongst the water, like you would find in the ocean or even in the bay that's a bit salty, the water in this case would be the solvent, and the solute would be the salt, the sodium chloride. Now, in the body, we will find specific types of solutions. For example, colloid. Colloid is a solution where the solute is a large organic molecule. And you'll know from the reading in the PowerPoint slides that I posted that an organic molecule is something that has a carbon skeleton. But in the body, we're particularly thinking, when we talk about colloids, of proteins. So the blood could be considered a colloid. It's a solution where you have the blood plasma, which is the water, and dissolved in it are lots and lots of proteins. Things like liver proteins that are there to help with clotting, or enzymes, hormones that are made of proteins. Some hormones are made of proteins, not all, some are. Various other proteins that are found in the blood. So blood would be a colloid. You'll also find it in certain glands, like your thyroid gland, where you're making a protein-based hormone, and you're doing that in a solution. So it's a colloid. There's proteins dissolved in the solute, so or in the solvent. So that's what a colloid is. Now, a suspension is when the solution has the solvent essentially settling out to form something called a precipitate or sediment at the bottom, and that would be that stuff down here. So here in the solution, everything is all interspersed and just floating around evenly. But in the suspension, some of those solutes have come out, and they're at the bottom. And I put a pretty thick layer here. Well, a medium layer, I guess you could say. It could be less, it could be more. And again, just go back to that glass of salt water. Think about if you got it from the ocean, here's the salt water, everything's all interspersed. And then let's say you added more sodium, or more sodium chloride, I should say. And it ends up being more particles, more solute, than the solvent can actually suspend. So it's almost as though the solvent is saturated, it has too much. So some of that solute's going to fall out and give you this precipitate at the bottom. Okay, so in the body, when we're looking at solutions, we need to think about where fluids are found in the body, and we're going to zoom in on a tissue and on one particular cell in a tissue. So this would represent the tissue. Here's a cell. There would be other cells all around it. The fluid that surrounds the cell is called the interstitial fluid, and that is part of the ECF. And the ECF stands for extracellular fluid, which just means any fluid that's outside of the cell. That's in opposition to the intracellular fluid, which is the fluid that's found inside the cell, or the ICF. And that would be the same thing as a cytoplasm. So cytoplasm is intracellular fluid. So I'm just going to use the abbreviations ICF for intracellular fluid, meaning inside the cell, and ECF for extracellular fluid, meaning outside the cell. So when we're comparing the intracellular fluid and the extracellular fluid, we're really looking at what's going on across this cell membrane, the semi-permeable cell membrane, which means that some things can cross the cell membrane, other things can't. Water can move freely across the cell membrane. Ions like sodium or potassium, those things require a transport protein on the cell membrane, or a carrier protein. I'll just put ion channel here for that ion to move across. So it's not that whatever is in the extracellular fluid and this interstitial fluid here is going to end up in the intracellular fluid or vice versa. There is a bit of a barrier, but they have a direct effect on each other because water can move freely. These ion channels, there are lots of them, lots of transport channels for ions to move across the cell membrane, whether going into the cell or out. And we're going to focus a lot on that when we get to the end of the semester and start looking at neurons and things like that, action potentials, electrical charge across the cell membrane. We're going to get really deeply into that. But just for now, just really remember, really put this in your mind, that ions can move across cell membranes with help. Water can move freely across cell membranes, and it'll be in response to a couple of things that we're going to talk about in a second, or briefly introduce. Before we get there, though, I do want to point out that the interstitial fluid is not the only example of extracellular fluid we have. We also have blood, that's outside of the cell, that's extracellular fluid, and there's lymph, which is, again, outside of the cell, so that is extracellular fluid. And if you just think about water, just imagine yourself taking a drink of water. It goes into your stomach, it goes into your intestines, it gets absorbed from those places into the blood. Now you have this water in your blood, so we'll just put some water there. And that water is going to move using osmosis. So according to osmotic forces, which is where we're going, but just bear with me for a second, it's going to move out of the blood into the interstitial fluid. Now it's part of the interstitial fluid. From the interstitial fluid, it can move into the cell freely, or it can move out of the cell freely in response again to osmotic forces, which we're going to look at in a second. So water will be moving in and out. Hopefully it's in an equilibrium situation, so it's moving in or out at the same pace, or we'll have a situation where we're either putting too much water in the cell or pulling too much water out of the cell. And we don't want that. We want the cell to have the same amount of cytoplasm, just a nice normal amount of cytoplasm. But the water's going to go in and out of the cell, back into the interstitial fluid. Some of it gets reabsorbed back into the blood. Some of it ends up going into the lymph, and the lymph has an immunological function, so it's carried in lymphatic vessels that just carry this, essentially what used to be interstitial fluid, through a variety of lymph nodes where you find white blood cells, specifically T cells. We don't need to get into all that, but they'll filter the lymph, trying to get rid of pathogens, killing any viruses or bacteria that have gotten into the interstitial fluid and ended up in the lymph that way. And then the lymph will eventually end up back in the blood. And then the water can continue, going back and forth among these compartments of extracellular fluid, intracellular fluid, and so on and so forth, or be excreted. Okay, so what's making things move across this cell membrane? That is a semi-permeable cell membrane, which, remember, that means some things can cross easily, some things cannot. So let's just set up two little beakers next to each other, divided by a semi-permeable membrane, like what you would find with the cell. So we know that water can pass freely, but we're not looking at water yet. We're just going to ignore water for a minute. We're going to look at sodium. Sodium cannot pass freely across the semi-permeable membrane in terms of a cell membrane. It requires a protein channel. So I'll just put this little protein channel, I'll call it a sodium channel here because that's being more specific. So it can cross using that. Now in lab you're going to be using a different kind of semi-permeable membrane, or you're going to be watching someone else, I should say, use a different kind of semi-permeable membrane where ions would be able to move freely. They wouldn't need a protein channel because we're just trying to recreate the situation there. So what the sodium would be moving in response to would be various gradients. Together the gradients are called the electrochemical gradient. And it's just the sum of its parts. There's an electrical gradient and there's a chemical gradient for an ion. The electrical gradient has to do with positive charges being attracted to negative charges. We saw that concept when we were looking at ionic bonds in an earlier lecture, but it's going to be something that happens within your body all the time. Positive ions like sodium are attracted to negative environments. And just for future reference, the cell, inside the cell, tends to be a negative environment, which makes sodium really attracted to want to go inside the cell. That's something that we're going to build on later, so just keep that in the back of your mind. And when I say later, I mean later in the semester. It's not so important right now. What we're really going to be focusing on right now is the chemical gradient. And that's where substances move from a high concentration to a low concentration. And that's what you're going to be seeing in lab, things moving from where there's a lot of them to where there's not so many of them. I like to think of this as sort of like when you go into a classroom, if you remember those days of going into a classroom, which we will have again, hopefully sooner than later. But just imagine yourself all filing in on the first day, going into the classroom, and just think of yourselves then as the solutes. Each one of you is a solute, and the environment of the classroom is the solvent. You're not all going to go bunch up in a corner together. That would be very strange, right? You're going to spread out in the room. Or imagine it in an elevator. You know, you don't go all stand in the same corner of the elevator when there's like 5 people on the elevator. You spread out and you kind of, people really evenly space themselves on an elevator. So if you think of the ions as sort of doing the same thing, they're trying to spread out as much as they possibly can. So when they have a situation like this, where we have a lot of sodium over here on this side and there's no sodium over on this side, the sodium is going to want to move in this direction. It's going to be pulled using diffusion to push over in this direction. We would call it diffusing over into the direction from the high concentration to the low concentration. And eventually what would happen is, in this situation, we have 4 sodiums over here, there's no sodiums over here. We would end up having 2 of them move over here, so that we have 2 sodiums here and 2 sodiums here. And then we're in equilibrium. So sodium would now be in a chemical equilibrium with itself, and it would no longer diffuse. That's not to say there'd be no movement of sodium, it's that there would be no net movement of sodium. So we'll say no net movement at equilibrium, which what that means is that if this sodium comes back over, then it'll either go right back in or one will go in to replace it. So they'll be moving back and forth at an equal rate. So we'll end up with, on average, 2 on each side. So how do we apply this to our body and water movement? So think again, just here we have our cell and we have the interstitial fluid surrounding it, so that would be like the semi-permeable membrane. Now inside the cell we have typically a lot of potassium, so I'll just put 1 potassium there. We have a lot of sodium typically outside the cell as compared to inside the cell. Now this is a semi-permeable membrane. These ions need ion channels to cross, but there are those ion channels, so they are able to cross in response to their electrical, their electrical and chemical gradients. And they're going to do so until they're in equilibrium. Now it's not quite so simple because we don't have just sodium that we're dealing with or just potassium. There's a whole bunch of other factors here. So we just kind of want to look at, for now, we want to look at just sodium and how it affects water movement from the interstitial fluid into the cytoplasm and vice versa. So we're just going to focus in on the cell and the interstitial fluid and how sodium in particular is affecting the movement of water across this channel or across this membrane. We'll worry about sodium movement across the channels and potassium movement across the ion channels later in the semester. So let's look at just the concentration of sodium in the body, in the interstitial fluid. So here's the cell. We're just going to focus on this middle one for a minute. This is the cell. This is the interstitial fluid outside of here. And this is, we're just going to call this a normal amount of sodium in the interstitial fluid. This would be the typical imbalance equilibrium situation. And we would measure this in what we call tonicity. Tonicity has a lot to do with the osmotic force, the osmotic pressure. That has to do with the movement of water across that cell membrane, whether the movement of water is going to be into the cell or out of the cell, or if it's going to be in an equilibrium where the water moves at an equal pace both in and out. And what we want is a situation where we're in equilibrium. We don't want water going into the cell too much. We don't want water being pulled out of the cell too much. We want water to just be kind of moving in and out at the same rate in equilibrium. And that's called an isotonic situation. And that's what homeostasis is going to be attempting to achieve for your body at any given time. That's why when you don't have enough water in your body, you get thirsty. Or if you have too much water in your body, you would excrete more water through mostly urination. Trying to maintain this isotonic situation. And we can also do that by manipulating how much sodium we have in the body, because sodium has a really direct effect on this osmotic force, and that's why I chose it as the example, and also because sodium is going to be the ion that you're using for one part of the lab. When you watch those lab videos, you'll see what I mean. There's some solutions that have sodium that you're going to be putting a cell into. Okay, so consider this cell. Normal situation with sodium. I just put 4 sodiums there. That's, just a made-up number. I put potassium in just to remind you that there's more potassium inside the cell than out, because that's going to become important later in the semester. So just getting used to putting that potassium inside and the sodium outside. So here we're in osmotic equilibrium. The water moves in and out at the same pace. So the cell is happy. Everything is fine. Now let's mess with that a little bit. We're going to put a whole lot of sodium in the interstitial fluid, and that's called a hypertonic solution. So the extracellular fluid, the interstitial fluid, has become hypertonic, and it's as a direct result of all this sodium. It could be other electrolytes that are causing it to be hypertonic. In our example, we're just going to use sodium. So what happens here is that the water gets pulled out of the cell, so the water is essentially being sucked out of the cell, and the cell's going to start to shrivel up. And if this is extreme, the cell can shrivel completely like a little raisin, essentially, and that is called cell crenation. And that will kill a cell if it's extreme enough. So you don't want that to happen. How that would happen is if you were dehydrated, if you were to drink salt water from the ocean. That's why they say if you're stranded at sea, don't drink the salt water, no matter how thirsty you are, because that will make you hypertonic and it will cause cell crenation. Among other things, it will also make sodium move into the cell faster, which makes cells hyperexcitable, which is also not a good thing. That's something we'll think about when we get into the end of the semester. But just from the point of view of osmotic force, crenation, not a good idea for your cells. Now, just sort of as a little fun example, and because people are pretty much always dehydrated, for the most part, I don't think hardly anybody drinks enough water, but there is a test to see if you are dehydrated and therefore maybe a little bit hypertonic. So you're gonna, I hope this isn't gross to anybody, but it's a really easy test. You just hold your hand out like that, take the skin on your finger above your knuckle and give it a pinch, and then let it go, and see how mine is not going back. I am dehydrated a little bit. Yuck, that's gross. So try it on yourself. If your skin doesn't, it should just smooth right back out again. If it doesn't, you're a little bit dehydrated, you're kind of hypertonic, you might be crenating your cells somewhat. Go drink some water. Okay, but the opposite situation, if you drink way too much water, and this is way too much water, like a gallon of water in one sitting, you could become hypotonic, or you could maybe never eat sodium, although that's pretty difficult to do. Especially with if you eat any processed food at all, you're pretty much getting all the sodium you need for a week with just one serving of that. So it's a little bit more difficult to become hypotonic. But that's a situation where you say don't have enough sodium or just not enough electrolytes. So you have like a low osmotic force, a low osmotic pressure. In this case, water is going to move the opposite direction. It's going to be moving into the cell, and it can cause the cell to burst. And that is called cell lysis. And that is also not good. If your cell explodes, that's obviously not good. Now I want to also note that these terms are relative to each other. So where you have a hypertonic situation, that means that it is in opposition to or in comparison to the fluid inside of here, which would then be hypotonic. So we're talking about the extracellular fluid. We would say, ooh, you are hypertonic because your extracellular fluid is hypertonic. But it's comparing it to what's going on inside the cell, which would then be the opposite, hypotonic. Opposite situation for here. Your extracellular fluid is hypotonic, and this can also be called water intoxication. But the extracellular fluid is hypotonic, so you would say you're hypotonic, but that's in comparison to what's inside the cell, which then by default, because it's opposite, it would be hypertonic. So if you look at the way the water is moving, when the extracellular fluid is hypotonic, it goes into the intracellular fluid, which is hypertonic. It moves out of the hypotonic solution and into the hypertonic solution. Same thing here. Remember, this is going to be hypotonic inside here. If you're hypertonic in the extracellular fluid, water is moving out of the hypotonic solution into the hypertonic solution. We can do it, oops, there, with our little beaker separated by a semi-permeable membrane. I'll put a ton of sodium over here. Let's say this membrane is not permeable to sodium, so the sodium can't just diffuse across. We'll put no sodium over here. That makes this the hypertonic solution. This is the hypotonic solution. Water is going to go that way. Just think of water as being really attracted to electrolytes, and that'll help. Or you can think of hypo, water moves out of the hypotonic solution towards the hypertonic solution. So hypo, there's that O in there, water moves out of that. Okay, so hopefully that should get you ready for the lab exercises that you're going to see. So after this video, watch those lab videos, read along in your lab books with that, and that should make sense when you put all of that together. Okay?